Proactive, precision, whole health medicine is the next frontier in healthcare. Our next panel will discuss how advances in technology, lifestyle medicine, and preventive therapeutics help not only prevent chronic conditions like heart disease and diabetes, but also maintain cognition into older years, because it's never too early to talk about brain health. How are the latest approaches and cutting-edge modalities giving health professionals an edge against Alzheimer's? Let's hear from our next panel, led by Julie Wood, Senior Vice President for Research, Science, and Health of the Public at the American Academy of Family Physicians.
Hello, thanks for joining us today. Precision medicine, sometimes known as personalized medicine, is an innovative approach to tailoring disease prevention and treatment that takes into account differences in people's genes, environments, and lifestyles. The goal of precision medicine is to target the right treatments to the right patients at the right time. We have an exciting panel today of experts to discuss the latest advances. Their bios are available on the website, and we invite you to take a look, but we're going to jump right in to talk about the important treatments and innovations today. Let's start with Dr. Kivipelto. Your research is in many ways the foundation, the building blocks of what we think of as Alzheimer's prevention or risk reduction, depending on how far one takes the science. Where would you start us off with an overview of the FINGERS trial? You have found and are finding what is important for medicine.
Thank you so much. My great pleasure to be here. Indeed, the FINGER trial was the first randomized trial to show that it is possible to prevent or reduce the risk of cognitive decline. And the concept was the multi-domain intervention. We put together several modifiable risk factors, and that's the lesson we have learned. It is maybe not enough to only target one of the risk factors, but you need to target several risk factors and mechanisms at the same time. What are those risk factors? It's easy to remember. The finger model is like one hand and 5 fingers, healthy balanced diet, physical activity, cognitive training, social activity, and taking care of all vascular metabolic risk factors. All what is good for the heart is good for the brain. And the results after the FINGER trial, first we targeted persons who had increased risk for cognitive impairment and some modifiable risk factors for 2 years compared to regular health advice. After 2 years, this group who received finger interventions actually improved cognition for different cognitive subdomains. And the cognitive decline was decreased by 30%. On top of that, this finger group had better functional level, better quality of life, reduced risk for stroke and multimorbidity, and even we have been able to show health economical benefits. So this is really not only for the brain health, but for the total. And what is nice now is that the finger model has been expanding to worldwide fingers in 60 countries. And here we are adapting, further developing the model. We are upgrading it to finger 2.0. And this is the personalized precision medicine where we try to tailor the intervention even better, taking into account individual risk factors. And in the future, combination therapies combining lifestyle and different type of treatments. So very exciting time indeed. And maybe one more take home here. I normally say it's never too early to start to prevent dementia, but we have also learned it's never too late. We have conducted a finger type of trial among 80 patients, very good results. So the effect of the intervention is not ending after the diagnosis. It may be even more important to take care of these different risk factors.
Great, thank you. Dr. Phillip, let's go to you. We know that ADEF stands for Alzheimer's Drug Discovery Foundation. How does precision prevention fit into the picture? What kinds of prevention research are you supporting? Why and how do biomarkers fit into this equation?
Thanks for having me today. Well, right off, I want to follow on what Mia said. We have a partnership with Mia to do what we've called together the Finger 2.0 study, which is now, I guess, the MET Finger study. And MET stands for metformin. Metformin is one of the leading anti-aging drugs, so to speak. We had funded a clinical trial of metformin for people with Alzheimer's a number of years ago, and it showed some promising results. And that investigator just got a $20 million grant to do a large trial of metformin for the treatment of early Alzheimer's disease in patients with MCI. But we think that, I should say, me as a group and our group together think that that it's kind of like the model that we have now in the prevention of heart disease, where we have all these lifestyle interventions to prevent heart disease. But through biomarkers, namely cholesterol, we know that cholesterol is a big risk factor, and that statins can play an important role in addition to lifestyle interventions to delay the onset and prevent heart disease. And so we're adopting that same sort of model where we would have lifestyle interventions, comorbidity management of diabetes and hypertension, the five fingers that Mia mentioned, plus drugs that are relevant to slowing down the course of cognitive decline in people who are aging. And I think biomarkers play a critical role. It's a very big part of what we do. We have a $100 million partnership with Bill Gates and Jeff Bezos and David Dolby and a number of other philanthropists to develop novel biomarkers for the early diagnosis and monitoring of treatment in Alzheimer's disease. I would say that if you think again about heart disease, imagine trying to develop drugs for heart disease if you didn't have a blood cholesterol. And in fact, the whole field of that prevention started when it was recognized in the 1950s that cholesterol was a risk factor for heart disease from epidemiological studies. It took about 30 years from that initial discovery until the first statins came to market. And what we're seeing today in Alzheimer's disease is also extremely exciting. We first funded the very early stages of the development of the PET scans that can detect amyloid in the brains of living people. And I know most of my patients didn't want to, at that time, and certainly now, go to autopsy in order to get a definitive diagnosis. And so we started funding that at the University of Pennsylvania back around the year 2000, which is about 25 years from now ago. And just to give you the kind of timeline of how this goes, in 2005, the Penn spun out a biotech company called Avid Radiopharmaceuticals to develop and commercialize the PET amyloid scan. In 2010, Eli Lilly bought that company for $800 million in milestone payments. And in 2012, that PET scan became the biomarker as the first diagnostic for Alzheimer's disease, went to the market. Medicare declined to pay for it because they said there was no point in making the diagnosis because there was nothing you could do. Of course, they didn't know about the finger study at the time. And then around 2014, it turned out one of the obvious reasons that Lilly bought this biomarker, neuroimaging biomarker, was because they actually wanted to see how many people in their clinical trials at the time of a drug called solanezumab, which is an anti-amyloid antibody, actually had the target of the drug, which was amyloid in the brain, and actually had Alzheimer's disease. And it turned out that about 30% of the people enrolled in those trials didn't have amyloid in the brain, they didn't have Alzheimer's disease, and it was probably one of the reasons why many trials up to that time with monoclonal antibodies to Alzheimer's had failed. And so what Biogen and other companies did, Lilly, is they started using that same test to make sure that the people who were enrolled had a positive amyloid scan. And long story short, the first disease-modifying drug ever approved by the FDA for Alzheimer's disease was based on what's called an accelerated approval, which basically said that because we could show with the PET scan that the drugs worked in doing what they were supposed to do, which was removing the amyloid from the brain. And so people that had positive amyloid scans at the beginning of the study had negative amyloid scans at around 12 to 18 months at the end of the study. And so the FDA concluded that there was a reasonable degree of certainty that the removal of amyloid would result in cognitive, the slowing of the rate of cognitive decline. And so the first approval of a disease-modifying agent was based on the test that we had started funding 25 years before that, and it was a biomarker. And the last part of the story is that I've been using that biomarker in my clinical practice, but my patients can afford to pay for it. It's quite expensive. But just this past week, Medicare, because these clinical trials were positive, showing that the monoclonals could remove the amyloid, Medicare has agreed to finally start paying for this biomarker, which I think is a huge advance. And I don't want to take up too much time here. I know I'm talking a lot, but I will say that the next stage of all this, real briefly, is going to be blood tests. We have them already on the market. A blood test for amyloid, blood test for tau, and blood tests for other aspects of the disease, which will lead to precision medicine. And I could be happy to talk about more about that. But again, I don't want to take up too much of the time here. So thank you.
Thank you so much, Dr. Phillip. Dr. Tanzi, most of the conversation in the medical community is around possible pharma treatments when some symptoms have already begun to emerge. Can you give us a glimpse into why what Dr. Kivipelto and others are doing has implications for asymptomatic prevention and why physicians should be paying attention?
Well, I think, the first thing is the elephant in the room is that we don't diagnose Alzheimer's disease until the brain is already deteriorated to the point of dysfunction. If we diagnosed heart disease the way we diagnose Alzheimer's, we'd wait until you had congestive heart failure rather than start treating you with cholesterol drugs decades before. And we have to make that shift. And I think, between what Mia's talking about in prevention with lifestyle, which is the fastest, easiest way to do this for your whole life, and Howard mentioned the cholesterol analogy. And I think the cholesterol analogy is perfect because the way Brown and Goldstein, who got their Nobel Prizes for discovering the cholesterol link, the way they did it was they had early onset families, early onset heart disease with a guaranteed onset due to a rare gene mutation in the LDL receptor that increased cholesterol. And like Howard said, it took decades to realize cholesterol is something you prevent you manage to prevent heart disease. You don't wait till somebody needs a coronary bypass to say, here, take some Lipitor. And look at how we discovered amyloid and Alzheimer's disease. We and others discovered early onset families. I did my first genetic linkage study in an early onset family in 1982. And then by the 80s and 90s, we had 3 genes with rare mutations that guarantee early onset Alzheimer's disease that all said amyloid, whether the mutations were in the substrate, APP, or the enzymes gamma secretase encoded by the presenilins, it all said amyloid. And we had to come to the realization that treating amyloid in a full-blown Alzheimer's patient was just too late. And it's something you really want to do pre-symptomatically with drugs that are safe and affordable, that can be democratized for care. Now, people differ on the answer to how many people in this country, in the US right now, don't have Alzheimer's symptoms, but already have amyloid accruing in their brains. And I get answers of anywhere on the low end from 10 million to the high end of 40 million. But in any event, that's tens of millions. So at the end of the day, we're going to need drugs that are affordable and safe that we can use early and often to hit amyloid as soon as we know it's accumulating. And one last thing I want to say, and I appreciate the idea of using a PET scan to know you have amyloid before symptoms and treating then. That's okay for now, but in the future, you have to realize once you have a brain full of amyloid, you've already triggered tangles, tauopathy, and those tangles are propagating. So you may clean the amyloid out very effectively with an immunotherapy like Leqembi or Aduhelm, but the tangles are off and running. So optimally, you'd want to hit the tangles at the same time, and more optimally, you'd want to have a drug that hits amyloid production before the head is full of amyloid. Turn off the tap. Don't just clear the drain in this sink that's overflowing with amyloid. So, I'm working on drugs like gamma secretase modulator and other repurposed drugs that are meant to not only clear amyloid, but I think more importantly, prevent amyloid production before you already have the disease process, beginning with amyloid accumulation and tangle propagation starting. And that's going to be a whole nother challenge. I think Howard's going to have to fund the next company that finds the biomarkers to say, you don't have any amyloid yet, but you're on your way to having amyloid. You need a drug that brings your amyloid production down. But that's going to be the next level after the current successes we've just seen over the last year or two.
Yeah, if I could just jump in, I agree with Rudy completely about precursors to amyloid and things like inflammation, which I'm sure we'll talk about. But one thing I didn't say explicitly is that these brain scans showed us that the disease starts maybe 20 years before people get symptoms. And so there have been clinical trials already identifying people in the community without symptoms who have Alzheimer's disease, as Rudy mentioned, based on the biomarker. And they went into treatment trials. And so far, those prevention treatment trials, which are classic prevention treatment trials, haven't shown us the light of day, but I do think that they will. So one of the biggest advances in prevention has been showing that we know the disease starts about 20 years before people get symptoms, and we can start treating those people and reduce it's been modeled that if we could reduce if we could delay the onset of Alzheimer's disease by about 5 years, we would reduce the number of cases by about 50%. Yes. And I think with what we know today, that's a very achievable goal.
Mm-hmm.
Let's go to Dr. Van Hecke. Dr. Phillip mentioned biomarkers. You have a company that's using technology, data, AI, and this is to predict disability and treatment by tracking disease activity and progression. Talk to us some about how technology, the way we're able to track not just brain disease, but actually brain resilience. Do you see a future where we can use technology to help us see improvement and the kind of things that result from this type of precision medicine interventions that Dr. Kivipelto is researching? How can technology help us apply these interventions sooner with more precision today?
Thanks a lot for the great question, Julie. It's also an honor for me to be on this panel. More than happy indeed to share a little bit of our experience at Icometrics applying artificial intelligence technology, and mainly in our case to brain MRI scans. I also very much like the analogy with cardiology, the role of biomarkers, the need for data for decision-making and to go to precision medicine. Also in diabetes oncology, we've seen that movement, and I think especially in neurology and Alzheimer's disease, there's a huge need there. Now, when we look at MRI, it offers a unique window to the brain. MRI scans are non-invasive, have typically a good availability in most countries. However, despite the wealth of information that is present in the MRI scan, the end result, which is a radiological report, is typically descriptive, qualitative, based on a visual assessment by the radiologist. And so it doesn't really contain biomarker measures or data or comparisons to population as clinicians are used to by receiving from blood lab tests or genomic analysis. And that's why at Icometrics, we have developed artificial intelligence solutions that can quantify clinically meaningful measures based on standard clinical brain MRI scans. For example, we measure hippocampal volumes, the number of small brain bleeds or microhemorrhages, the frontal, parietal, occipital, temporal lobe volumes, but also compare them with a large population to also try and differentiate early pathology from what we can consider as normal aging. And these measures are FDA cleared for the assessment of individual patients, not just in studies or research analyses. They are relevant to diagnose Alzheimer's disease early, of course, in combination with a lot of other biomarkers. And of course, with new disease-modifying treatments becoming available, MRI measures are also relevant to assess eligibility for these drugs. Is a patient eligible to go on one of these new DMTs, but also to monitor treatment efficacy and also safety. And there on the safety side, we've all heard about amyloid-related imaging abnormalities or ARIA. We developed an AI solution that can actually assess ARIA, brain bleeds, and different types of side effects, which is FDA pending. And this will become very important. Up to 35% of patients are suffering from ARIA when they're on amyloid therapies. According to the FDA label, you need at least 4 MRI scans in the first year to really monitor and track ARIA and quantify it. But ARIA is new to most physicians. It can be extremely subtle. It needs counting of lesions, which is already very hard to do with the naked eye. And it has been published that a very large amount of ARIA cases can actually be missed, especially by local radiologists that have not been part of clinical trials. So AI software will be very important, especially now with these new treatments becoming available to assist radiologists and clinicians and really getting data and measures out of these scans, in addition to the radiological report and the visual assessment. So that's kind of what we do at icometrics. It's about giving physicians access to measures and data on the MRI scan, the data that they need to optimize care, allow for more personalized and precision medicine. And maybe one final comment also, as you also mentioned, brain resilience and how MRI could play a role in seeing improvements that result from precision and preventative medicine approaches. Our brains are famously flexible, and because of that brain plasticity was already mentioned, we can cope with fundamental pathological changes for a long time before we experience symptoms. And also there we have quite a lot of experience in Alzheimer's disease, but also in other chronic neurological conditions such as multiple sclerosis, where we see that brain scans are typically very predictive for future disease onset or progression, even for therapeutic response. We're also developing some ways of looking at the MRI and indeed predicting amyloid burden in patients. And so that's really ongoing research. And MRI is not only relevant for diagnosis and monitoring, but could indeed also play an important role when we look at that prevention and prediction in the future.
Great, thank you. Dr. Walker, your intervention also, you're welcome. Your intervention also uses data and technology, but also blood samples and genetic tests, correct?
Yes.
What other information do you feed into your platform, and what treatments result from that? How do we know it's working? And what else are you doing that aid our collective efforts to stop Alzheimer's before symptoms set in?
Sounds good. What we do is we look at the treatable causes of cognitive decline, and those are kind of well known, and there's dozens of them, and pull those together for a family physician. At the moment the patient's standing there, those are called care plans, and it's a stepwise process of how to treat this person, what to measure next, and where to go. I'll use a word that you all know. It's translation. We're a translation company. We're not doing research. So that means we're dependent on 30 decades of, the best evidence-based research studies and trials. And I'm going to call out kind of by name that says, everything in the finger recommendations and the follow-ons is in our care plans. Dr. Kivibelta, thank you. I see Dr. Phillett, everything in your wonderful supplement database, influences some way how we recommend supplements or not. Dr. Tanzi, everything in genome-wide, the GWAS influences our direction. So we've talked about several inputs here. How does this all happen in real life? And what happens is a doctor can order a single blood panel. It's already in the ordering systems for Quest and some other labs. And that triggers a cascade of events. But, as a business, how does this happen? When the blood is drawn, we get the raw data, and it's about 100 biomarkers. But mostly those are simple biomarkers because we're trying to address the multi-domain issues of what things are going on all at the same time. When that blood data, raw data arrives to us, then that also causes us to now go and follow up with the medical history of the person. So we can see electronic medical records and so on. So now look at the picture that says we've got current blood data, we've got history in their medical records, we've got their problem list, their medication list, their allergies, we've got genomic data, we've got imaging if that's available, we've got amyloid markers, and all of those together start to form a picture of what to do with this person and how to proceed next. Let me mention a couple of things, and they're the key words from what we see. We've looked at about 10,000, more than 10,000 people at this point. And those two words are multimorbidity and polypharmacy. For those who don't know those two words, multimorbidity is having multiple diseases at the same time, and polypharmacy is taking 5 or 10 medications, pick where the line is. 80% of the people we see have 3 or more chronic diseases. You can't really address the progression of Alzheimer's, even from the prevention stage, without addressing the issues related to the chronic diseases. And so that, again, becomes a prioritized piece of what we recommend. Second of all, we see people on 10 medications on average with a standard deviation of 12. Meaning we see people on 20, 30, 40, 50 medications a day. Quite astonishing. We call that a combinatorial explosion because they'll have 400 or 500 active drug-to-drug interactions. And until that gets unwrapped, you kind of don't make much progress through this journey. Let me talk about one more thing. I've got a lot of examples here, but I'll just pick on one here. 58% of the people we see have chronic kidney disease. It's very undiagnosed because only 13% show up with that in their reports. And it's because we've done a thorough set of looking at the biomarkers. Chronic kidney disease is a driver of Alzheimer's disease. So they've got chronic kidney disease. It's not been diagnosed. It's working away at the progression of Alzheimer's disease. And then the next thing we see is because we've got their medications, they're on 2, 3, 4 medications known as, and there's a term for this, nephrotoxic. It poisons the kidneys. There's a whole stat pearls of just the nephrotoxic medications. So that's part of the unwrapping we do in what we do here. I didn't answer your question exactly. This says, let me make sure I make a clear answer. Julie, we do translation. So we're dependent on the excellence of the people I see here and another 10,000 people around the world. And we, measure and compare. And so everything we've seen in terms of outcomes we have published.
Thank you. So I'm going to ask a similar question to Dr. Tansey. I understand your center has developed a similar sort of risk profile assessment. How is it being used now, and how can we use, or is it being used now, and how can these tools be used to measure amyloid risk, not just presence? And how do we prevent the brush fires, not just put the fires out?
Okay, in 3 to 5 minutes, let's go.
Yeah, let's do it.
At the McCants Center for Brain Health that I co-direct, we have what's called a brain care score. If you Google McCants, MCCANCE, and brain care score, and we ask things about your numbers, cholesterol, your A1C, your hypertension, your diet, your weight, your social habits, even things having to do with meaning in life, and it calculates, kind of a brain care score that we're using. We're trying to just generate as much of data as possible, correlating the brain care score with the state of folks in terms of pre-symptomatic disease or disease. But the real, the other side of the coin is very similar to what Mia said, which is, you have the 5 fingers, Mia. We used an acronym I came up with called SHIELD. Shield your brain, where sleep, S is sleep, H is handling stress, I interaction with others, E exercise, L learn new things, D diet. And it's an easy way to remember, what to keep track of during the day. But the big question is, how do we correlate life exposure, habits, just lifestyle with the biomarkers that are going to predict disease? And I said this before, and I say it again, we have biomarkers that predict the disease, that tell us the disease process has begun, even though you don't have symptoms. For example, the A-beta 42/40 ratio in the blood, the phosphatidylcholine levels,
Yes.
or if there's cell death, neurofilament light chain, neurogranin for synapses, et cetera. There's inflammatory markers. But all of the biomarkers we have now, the good news is they're pre-symptomatic. The bad news is that's not good enough. We need to know, we would like to know who's going to get into trouble. We want to know not just pre-symptomatic, but who's on their way to amyloid tangles, inflammation. Can we predict that from lifestyle? Can we start looking at biomarkers when people are perfectly well before there's any changes in the brain, and combine that with lifestyle data to say, we need to predict how you're doing. We need to give you a checkup from the neck up, as Patrick Kennedy likes to say. When we go to the doc every year, what do they do from the neck up? They look at the holes in your face, your nose, your ears, your eyes, your mouth. 3 pounds of jelly in there that matters. So that's the idea, is how do we get routine brain care? How do we get routine healthcare for the brain that takes into account lifestyle, and biomarkers that are even better than the ones we have today. As good as they are, they're telling us pre-symptomatic, predictions, but we need something that comes before the pathology that predates the symptoms, something that tells us pathology is on the way. And that's going to be the big challenge. And I think we got the perfect people on this panel, in the future to address this challenge.
So Dr. Phillip, how do we make sense out of all this as physicians? And I'm thinking with my primary care physician hat on, I work for my colleagues, family physicians. We have new technologies, biomarkers, interventions that are being developed, and every day in the news we're hearing about new things, which is great. But how do we look at the research and what's being developed and make sense of it for clinicians today to do a good job for educating physicians and other clinicians and take good care of our patients?
Yeah, well, I'm a geriatrician, not a neurologist, as it turns out. And I know Dr. Kivipelto is also. So when John Walker talks about doing seeing the kinds of patients he sees with, they're on 20 medicines and they have 10 diagnoses.
That sounds very familiar.
Yeah, that's just an average day at the office for us. So I think in some ways, what we're advocating here is quality care and, good lifestyle practices. And as Mia said, and John said, I think also what's good for your heart is good for your brain. And the hope is that that by saying that, we'll incentivize people to do the right thing. But I think the great advance that Mia, Dr. Kivipelto brought to the field is sort of in the realm of implementation science, because we know the average time that a doctor spends with a patient is about 8 minutes. And even if you're counseling the patient for those 8 minutes, they're going to go home and do whatever they want. So I think, if we can find a way in the health system to ensure that we know what to do, we know what the right thing to do and how to counsel our patients, but getting them to do it is the real trick. And I think that's where we need a system-wide change to try to enable doctors or the health system to provide this kind of preventative care, with the end result being prevention of Alzheimer's disease. And I do think the biomarkers can help us. So we have, for example, are developing something called the Speech Consortium, which we'll be announcing in a short time, because we're trying to find ways using the iPhone, for example, to pick up every very early stages of cognitive impairment. And we think that by doing that and bringing that to the attention of people, the earliest stages of clinical symptoms as expressed in people's speech, that we might be able to put that motivation in them to lead a healthy lifestyle and manage their diabetes and their hypertension correctly and aggressively, to do the right thing. So I think biomarkers will play a role. And what we're going to see as hopefully an addition to what the primary care physician, where most of this care starts, I would say, is to give them the tools. We had a big run up to get them to doing brief cognitive assessments. I think if you talk to a primary care physician today, they probably know what a MOCA is, which is a brief cognitive assessment. But 10, 20 years ago, nobody ever heard of that. And so, it's that kind of advances of what's practical in primary care that will ultimately lead to the application. I think we're going to have digital biomarkers of all sorts. We can do cognitive assessments of fairly in-depth using digital technology. There's a digital clock drawing test now that measures like 800 variables in the way somebody draws a clock. So I think, combining that with artificial intelligence and then making it available either direct to consumer or, in my opinion, preferably through through physicians adopting these practices so that they can help their patients get diagnosed early. And that can be the incentive. I also think the brain imaging, the quantitative volumetrics that Icometrics and other companies are doing can also be really helpful in identifying the earliest stages of neurodegeneration, and should be part of the early stage workup of patients just to see if there is neurodegeneration. And then the last thing I'll say real quick is blood tests. As Rudy mentioned, we're going to have a whole panel. They're already out there of blood tests that are going to not only identify patients at risk, but subset them according to this precision medicine idea. And then once we've identified these populations, we're going to be doing clinical trials in these populations and it's already happening, neural anti-inflammatories to reduce inflammation in the brain, and a whole host of other things. We're repurposing Ozempic, which is, already the most widely selling drug. But the interesting thing is that, it probably will work for Alzheimer's disease, because insulin resistance is a very critical part of the aging brain. And if the brain, which is 3% of the body weight and uses 20% of the body's energy through glucose at any given time, is starved for glucose because of insulin resistance, you're going to get neurodegeneration. And so it's really exciting that I think these GLP-1 agonists are being tested now. It's just one example of precision medicine. Do hemoglobin A1C, but now know that if a patient has that insulin resistance, they need to be treated aggressively, not just to prevent the typical diabetes complications, but also cognitive decline and dementia. And I could go on, but I'll stop there.
Hey, Julie, if I can quickly add, we have tools in the PCP office. You're the PCP. You have stethoscope. You have blood pressure. We have ways to assess heart health with simple devices. We don't have that for the brain. And I think, just taking off on Howard's comment about speech, eyes can also tell you a lot. I started a company called React Neuro that does a VR device. It does eye tracking, voice analysis, and gives you a virtual reality neuropsych exam at the same time, with the idea of assessing brain health and risk for brain disease. Someday, the hope is to have devices like this in your annual checkup. And because right now, there's nothing like that in the PCP office, but we have lots of that for cardio health.
Great, thanks. Dr. Kivipelto, what excites you most about the future? What is precision medicine for Alzheimer's prevention going to look like in 10 years?
Wow, it's so exciting time in Alzheimer research, both prevention, early detection, treatment, new technology. And I think when we start putting together these pieces, combining them, we really can advance the field. And listening all the speakers and panelists here, that's what is happening now. Prevention, I think, is the key. And this is both non-pharmacological and pharmacological, starting early, everything what we are doing. And here, there are so many things what we can do already today. 40% of all dementias, at least, are linked to modifiable risk factors. And as Dr. Phillips said, just postponing the onset would have a huge impact on the whole society. So here, I'm very optimistic for the future. If we could implement the knowledge available. It takes normally 15 years at least to start to implement the science what is available. I think for lifestyle interventions, at least, it's too long time, and we could try to do it a bit quicker. And how to do that, these tools, I believe new technology could also help if you want to implement finger type of intervention. So using new technology, all the tools and biomarkers, I think is really, really exciting. One more thing what I hope we can do is really tailor, as I said, the precision prevention. Who are the responders? What kind of advice we need to give to certain persons? When should we combine finger type of intervention with different drugs, repurpose or new type of treatments? We have some examples from finger studies. ApoE for carriers. ApoE is the most important genetic risk factor for AD. ApoE carriers seem to get more benefit from the intervention, finger intervention. We saw this in Finland, France, and now even in Japan with different kind of genetic background. We would like to study this more. And I'm also thinking, Rudy, I would like to discuss this more, how non-modifiable the genetic factors are. Can we do something with treatments? And even the age, we have been able to show that finger type of intervention can counteract the shortening of the telomeres. So we can really learn more about with the biomarkers, and that can help us identifying new targets for the intervention. So this new field, having the new biomarkers, new type of treatments, I think is really, really exciting.
Thanks. Dr. Tansey, you've been around this area a while. Do you concur? What does the next frontier look like?
Yeah, I think, just to answer Mia's question, I think most genetic risk factors can be overcome. When we talk about penetrance, the odds that a gene mutation will cause a disease, the real question is, will it cause a disease within a normal lifespan? So when lifespan was 45 years old, the early onset fully penetrant disease genes were risk factors, right? So it's always going to be that case. So even with the most severe fully penetrant genes, given that they're age-dependent risk, lifestyle matters. And
Yes.
it's always going to be a huge part. But I think the future, because we're running out of time, is early detection, early prediction, meaning family history and genetics.
Yes.
To get an idea of when you're going to be in trouble. Then early detection at the right time. For one person, you might want to start getting checked when you're 30 or 40 years old. For someone else, 50 or 60, depending on your genetics and the prediction of your risk. So that's the early detection. And then most importantly, early intervention. And now we, as clunky as it is, we do have an amyloid therapy. Yes, it's expensive. It requires MRIs, as Aria, as Wilm said. But it's opened the door. It's opened the door for safer, more affordable ways to do the same thing. If we can know amyloid's there, or better yet, even the amyloid's coming early on, stop that process, and you're going to wipe that slate clean. I think the genetics tells us that, and we're going to be in the same boat the nice boat that cholesterol and heart disease has brought us for cardiology health. So I think that's the future, is do for the brain what we've done for the heart, with early prediction, early detection, early intervention. And we're on our way.
Great. We have time for some questions from our audience, and we have several rolling in. So I'm going to turn this one towards Dr. Walker, who shared some interesting information. It says, what differences have you seen in your data for patients of color, specifically Black, Latinx, and American Indian?
We haven't, in that we don't use race as a marker. So we carefully look at genetics when we get those things. And the lesson is, everyone's pretty much of a mix of things from across the world. So it's more correlated with specific genes than it is with the concept we use culturally as race. I'll do a different one-liner that is culturally unfortunate in this country. That says, the single one number that's the best predictor of Alzheimer's disease is years of education. The single one number. And unfortunately, that appears to get matched, culturally in wrong ways, I believe.
John, you're right. I mean, when we do stratification by ancestry, we never use self-reported race, because
it's just not real.
but you can use genetic, whole genome sequencing would then tell you where you are ancestrally, and then we can start to subdivide that way. That's much more scientific way to do it going forward.
Yes.
And if I may add, I'm thinking the diversity aspect in all research we are doing is, of course, something we need to do more and more. I mentioned the worldwide fingers is now in 60 countries. Many of those are low middle income countries. So we aim to collect data, not only clinical data, but blood samples, genetics. I think that will be also a wonderful opportunity to study more biomarkers, gene environmental interactions, the cultural, socioeconomic aspects. So I think that's the direction we also need to have.
And this
Dr. Phillip used the word subset earlier. This seems to be the easiest subset we can do is those with APOE4. There's 25% of the population, and it's not matched to race, or it's not matched to other genetics. I mean, it's worldwide 25%. And in a panel discussion of prevention, that's the first place I would start, which is identify in 10-year-olds who has APOE4. It's inexpensive, and they need some urgency in their life to deal with the fact that they're likely to be getting this earlier. If they don't exercise, they don't do the right steps, and it's easy to identify that subset.
And if you have a safe drug that lowers amyloid production, it doesn't wait for amyloid to form and then clear it like we have now, then those are the folks where, if it's safe enough, you might start to get them on that drug early on, certainly for an E4 homozygote.
Yeah.
And one more addition, sorry, Howard, was the MedFinger study supported by ADDF is ApoE4 enriched population. At least 50% are having ApoE4, because that was the hypothesis that they need this kind of combination and intervention more than others.
But I think we've looked at ApoE4 as a therapeutic target see if we could modify the risk that's associated. And it turns out ApoE has so many biological roles that trying to target one of them is not a druggable approach to trying to fix ApoE4. And I should say that about 20% of the population has an ApoE4, either as a homozygote or a heterozygote, but 60% of the people with Alzheimer's disease end up having ApoE4. So it's a big deal. It's a low-hanging fruit. And you'd think that we've known about this for about almost 30 years now, and we haven't been able to fix it. So one way we're trying to fix it is actually with gene therapy, because we know that people that have ApoE2 have a reduced risk of Alzheimer's, and the ApoE2 offsets the ApoE4 risk. So imagine if we could put a virus into a patient's brain that was a non-infectious virus that carried the ApoE2 gene, so that the ApoE4 brain would make ApoE2, and that protein would offset the risk of ApoE4. And we were doing that. We spun out a company, or we participated in the spin out of a company from here in New York from Weill Cornell called Lexio, that's doing just that. They're using adenoviral vectors that have ApoE2, and they've shown in clinical trials already in phase 1 that it's safe and that you can see ApoE2 in the spinal fluid. So the next step is whether this sort of holistic gene therapy approach to this low-hanging fruit of ApoE4 as a huge risk factor for Alzheimer's disease can be fixed genetically. There's also been talk of using CRISPR technology. I don't know too much about that, but there's been other thoughts these days of how we could try to fix the ApoE4 problem through gene therapy. Probably Rudy knows a whole lot more about that than I do.
You just have to be careful because, with CRISPR, you don't want to have all E2 because then you have other issues if you have only ApoE2. You want a 2 or a 3, that's optimal.
So here's a question. What is your view about whether metformin should be taken to help reduce Alzheimer's disease risk? Do we know enough?
Can I say one quick thing about metformin? We did an unbiased screen of every FDA-approved drug in our mini human brain organoid Alzheimer's in a dish model. It's made screening very fast. And metformin, I'm not supporting it in any way clinically, but I'm just saying in the lab, in our screen, metformin hit the trifecta. It stopped amyloid from inducing tangles. It induced microglial cells to clear more amyloid, the way Leqembi does, and it also reduced the ability of microglial cells to be activated into a neuroinflammatory state. And that was an unbiased agnostic screen. So I don't take metformin yet, but I'm wondering whether someday we're going to call it vitamin M.
Yeah, and this is the question, when to start and what dose, and that's why we are conducting the MedFinger trial. There are a lot of pharmacoepidemiological studies on top of these experimental nice studies showing that people who have been using metformin seems to have a lower risk, but there are still need for strict randomized trials, and that's the reason why we have metformin, the MedFinger study, what dose, when to start, and what is the ideal target population. But I think it's very interesting drug.
Yeah, and I've prescribed it for patients, and then I started feeling uncomfortable because these people had normal hemoglobin A1Cs, and I thought, gee, this is maybe not the best way. So the way I've come about it is to measure people's hemoglobin A1C, and if it's 5.9 or 6, if it's in the range where they're sort of pre-diabetic, then all the more reason to prescribe it, because maybe it'll help your brain as well as every other organ. But as far as people that have normal hemoglobin A1C, I've become a little bit more reluctant to just go out and prescribe it, even if patients are consenting and informed. So it's, I think, a conservative approach to how it could be prescribed in clinical practice, with the idea that it might be neuroprotective in that sense.
How safe is it, Howard? Because it does seem to hit AD pathology separate of the whole A1C thing. I mean, how safe would it be?
Well, I mean, I think it's generally a safe drug, but we don't really know in people that have normal hemoglobin A1Cs what the safety profile. I mean, in terms of side effects, we know the side effects, but in terms of long-term safety in normal people, I don't think we know.
Yeah,
we know the answer to that.
Thanks.
How long do you think it will be before we see a blood test and a preventive precision intervention that is reimbursed for non-symptomatic patients?
Oh, dream come true.
Yeah, what we do is 100% reimbursed by Medicare for those who don't reach the threshold.
Can you dive into that a little more?
You need to arrive at your family physician with a poor score or existing MCI diagnosis or something before it gets reimbursed, unfortunately. Now, once that happens, then the whole process continues and everyone, it's a good process.
But that is the holy grail, Julia. A blood test that would say the disease process, the pathology, never mind symptoms, haven't even started yet. But we can tell you're on your way. It might be from genetics, like an E4 diagnosis, might be from biomarkers, but that's the dream come true, is to really nip this thing in the bud.
But we really don't know how long it's going to be. That's a little bit difficult to predict.
Well, we get the typical 5 to 10 years answer. Yeah.
Okay.
But now I'll go back to multimorbidity, because there's so many issues interrelated to, and I hate to pick on obesity and heart disease and lung disease and kidney disease and so on, that these are all diabetes, these are all related to this. And those need to be addressed in concert.
A lot of confounding variables. Could the zip code of where you were born and spend the majority of your life be a biomarker?
I do a quick yes, in that there's a lot of studies related to air pollution. And so every zip code you can find in the United States, its particulate load and air pollution load and so on. Those exposed to air pollution have more likelihood of Alzheimer's than those not.
Yeah. It could be indicative of education as well.
Yes. That is also the socioeconomic status and other factors. Air pollution, that's also a nice example of the more novel risk factors. It's not only individual, but also the environment. So we always think the interventions, we should have the individual and societal approach. Both are needed, making the healthy choices the easy ones in the future.
How do you describe the role of neural inflammation in Alzheimer's disease?
Well, without it, you don't get Alzheimer's. I mean, if you look at resilient brains, like Teresa Gomez-Easels looked at, meaning people who died in their 80s, no cognitive problems, and then to the surprise of the neuropathologist, full of plaques and tangles, the answer is always the same. Somehow plaques and tangles are there, but there wasn't a huge neuroinflammatory response at the level of microglia and astrocytes. And without that cytokine storm and neuroinflammatory response, you don't have the excessive cell death and synapse loss that you need to get symptoms. And so that's really a great clue. And it's no surprise that in GWAS, 60 of the 100 or so AD-associated genes are microglial genes that control that process of whether you're going to trigger neuroinflammation. So it's a key fact. I like to think of amyloid and tangles as the misfolded proteins that trigger the possibility for neuroinflammation, but it's only a threshold of neuroinflammation that will lead to enough cell death and synapse loss to get symptoms of the disease. That doesn't say neuroinflammation can't come early as well.
Yeah. And to add, I think it's fascinating if I'm thinking the drug development, AD drug development pipeline, like Howard mentioned, there are like 150 drugs or targets, and big part of these are now actually targeting inflammation. So it's clearly some of the new interesting targets for AD.
But it's also true that there's a relationship between systemic inflammation and brain inflammation. So we know that systemic inflammation primes these inflammatory cells in the brain called microglia to become inflammatory and express these cytokines, which we think, as Rudy said, are related to cognitive decline and the onset of symptoms from the amyloid and the tau. And I mean, there's a lot of evidence for that. There was one animal model where they took a typical transgenic amyloid model and made what's called a double transgenic, where they gave these mice osteoarthritis, and the mice that had osteoarthritis plus the risk of developing amyloid and developing Alzheimer's disease, they were much worse because systemic inflammation primes the microglia in the brain. And I think the clinical example of that is that when we get sick, like if we have a cold or if we have other kinds of illnesses, we feel bad and we're foggy, and that's called sickness behavior, which is the result of inflammation in the body affecting the mind essentially. So it's also part of the reason why I think managing diabetes and hypertension and exercises and so on reduces systemic inflammation and prevents some of that priming.
This is going to be our last question for our session. Dr. Tansey said lifestyle interventions are the best, earlier the better. So how can we improve the uptake of lifestyle interventions? Is this possible? That's a tough one.
Good luck.
Well, is it the carrot or the stick? You know, I mean, you can either scare people into it or you can tempt them with reward, but I think that's a huge challenge. I mean, Mia must have encountered this.
Yeah, and I think finger studies showed it's possible. People need support, they need knowledge, they need some tools. And always we think the finger should be something positive, showing the good direction. So having good feedback and education there is, but it's possible. We have been still so able to see it after 7 years, even 11 years after the 2-year intervention in the finger trial, that it's possible. And even small steps, you don't need to do everything.
See, the thing is, if you're on a diet, you can weigh yourself and be happy about the result, assuming you're doing well. We don't have a way to tell people when they're trying to improve their brain health, you're doing well. That's why we really need these easy measures that say, yeah, keep going. You know, you're doing a good job.
Yeah.
It's that whether that's the carrot or the stick, if people could know how they're doing with their 5 fingers or the shield, that would be an important part of it.
I agree, fully agree.
There's a company out there called Silver Sneakers, which is a managed care product that I was involved in many years ago to be developed, where a woman wanted to get older people to exercise. And everybody said, you're crazy. We can't get 25-year-olds to exercise. How are we going to get 75-year-olds to exercise? Long and short of it is she created a model where she realized that the people in the gyms, young people go to the gyms, you know, from 7 to 9 o'clock, and then they go to work, and then they come back and lunch. So the gyms are empty between like 10 and 11 o'clock in the morning. So she created a program. She became the largest contractor of gyms in the United States. She had ultimately 18 million people had this benefit called Silver Sneakers, where if you join a managed care like Humana, which used to own it, then you get a free gym membership. And when you go there, there's a geriatric physical therapist that helps you. You get to socialize, and they all go out to lunch after they exercise, and it's free. And they you know, so that's an example. I mean, she sold this little company for $100 million to Humana eventually. But I think it's an example where we can create system-wide changes that encourage the proper health seeking behavior.
But what do they eat for lunch?
Yeah, exactly.
They went out for lunch. They went out for lunch.
Okay.
Yeah, I want to say thank you to our panel. We're out of time. This was a fantastic, robust discussion and very exciting to learn about what we can help our patients with and our communities. And I thank you to all our panel and to all of us here today.